everybody. I would like to thank you so much for joining us today on this webinar about B-cell cloning. My name is Haley Dyer, and I'm the Marketing Director at Biosero. I'm going to be hosting the webinar today and keeping on top of questions that flow in. And so before we get started with our presentation, I just wanted to share some quick housekeeping information with you. The first important item to mention is that we welcome your questions, and you can submit those questions through the questions section on the GoToWebinar panel. So my plan is to hold the questions until the end, but you're welcome to add those questions in at any point. And the next thing I wanted to mention is that we're recording this webinar. So if you want to refer back to it after the event or share it with a colleague, it's going to be available for you. So without further ado, I would like to open the floor up to our speakers today. Matthew, could you get us started? Share a little bit about yourself and Cellenion.
Thank you, Haley. Yes. Hi, everyone. I'm Matthew Bennett. I'm the Chief Technology Officer here at Cellenion. And it's my pleasure today to introduce you to our technology, the Cellenion and the Cellenion HT, and to show you the great work we've been doing with our sister company, Biosero. I will start with a short introduction of our company, Cellenion. It's a spin-off of Cellenion, with a world leader in precision dispensing. It was established in Lyon in 2016, and the first Cellenion instrument was sold in 2017. Today, we have over 150 Cellenion installed worldwide, and we are available globally through Cellenion and its distribution partners. We are part of the BICO group since 2020, and we are now therefore working together with Biosero, which Dustin will introduce to you.
Yeah, Dustin, could you introduce yourself and Biosero?
Absolutely. Thank you, Haley, and good morning or good afternoon to everyone listening here today. So a little bit about Biosero. We develop science-centric software and laboratory automation solutions for researchers around the world. And so at Biosero, I'm an application consulting scientist in that role here. So we have over 100 employees today, over 750 customers around the world. We have 2,500 installs, and we're also in 15 of the top 20 pharma companies around the world. Founded in 2003, and we've been with BICO since 2021.
Awesome. Well, thank you both for joining us today. And with that, I think let's get this presentation started. Matthew, would you like to dive in?
Yes, thank you, Haley.
Okay.
So before we jumped in our solutions, I wanted to take a step back and look at the history of monoclonal antibody development and what the future entails. It all started in 1975, when Köhler and Milstein introduced the hybridoma technique, and therefore they reached the production of monoclonal antibody with predefined affinity. This was a breakthrough finding, and it revolutionized antibody research and paved the way for clinical advancement. And therefore, in 1984, they received a Nobel Prize for their work. In 1986, so only 2 years later, the FDA approved the first monoclonal antibody-based drug. Today, these drugs dominate the pharmaceutical market, and they are used in cancer treatment, to treat autoimmune disorders, metabolic condition, and infectious diseases. There is little doubt that they will continue to shape the future of diagnostic and therapies, and that they will play a key role in improving patient outcomes. When we talk about monoclonal antibody development, there are a number of methods that can be used. Here I've displayed three, maybe the three popular ones: hybridoma, phage display, and B-cell cloning. And today we will concentrate on B-cell cloning. B-cell cloning has some advantages and disadvantages over the other methods, and they are used in specific application based on these advantages and disadvantages. So advantages are they are better suited for therapeutics. The speed of development of these antibodies is very interesting, and the diversity of the antibodies that can be produced using this method is also a plus. In terms of downside or limitation, the complexity of the method is one, and it's expensive. So it's both in terms of equipment and running cost, because you need skilled operator to do it, and you have a limited availability of samples. The workflow in B-cell cloning is as follows. When you use B-cell cloning for monoclonal antibody development, you start with B-cell isolation, and this is where we're going to focus on today. It's followed by a screening, selection, sequencing, vector construction, transfection, and followed by monoclonal cell line development prior to production. There are at least 2 steps in this workflow where single cell isolation occurs. One here on top at the B cells isolation, where a high throughput is needed so that you can screen a lot of antibodies. And here at the end, so that you select the few colonies that are the most promising for your production. And today, again, we're gonna focus on this first step up there. What happens up there? First, blood is drawn from an immunized animal, and PBMCs are separated from the blood sample, and then the B cells, and then B cell panning occur, followed by which the single B cells are isolated in the microwell of a microtiter plate just prior to screening. This step here is typically performed by a FACS, and what we've heard from people in the field from our prospective customers and users of FACS is that there are important pain points that we address with our Cellenion technology. These pain points are first in the category of the running cost. The FACS requires a skilled operator to be present at any time it is running. So it's skilled labor intensive. So if you run the FACS for 4 hours, you need 4 hours of scientist in front of it, or technician. It requires consumables for calibration and for running. And the running cost is increased when you want to have a good manufacturing because you have downtime for maintenance. Second big category that is a pain point in using the FACS is a throughput. Typically, we are being told that shear stress might be an issue that affects cell viability, and therefore you don't obtain as many colonies as you wished once you've isolated these cells with your instrument. Second point is because the FACS is operator intensive, so you have a lot of manual operation when you want to run it to reach an important throughput. And this is because you need to exchange sample and target manually, you need to troubleshoot manually, or you need to calibrate manually. We took all this consideration into account, and we think, or we know actually, that our technology answers all this limitation of the FACS for B-cell cloning. What is our technology? It's called the Cellenion, and I will show you some videos of how it works later, but basically what it does is that you can reach up to 100% single cell accuracy with it. It's an image-based sorter, so you can select with criteria and you see the cell that has been isolated, therefore giving evidence of monoclonality. The images are saved. You can select cells based on criteria. It can be shape, it can be size, it can also be fluorescence. So you can, for example, make live cells gating. You have seamless adoption because we don't have consumables. So you use whatever consumable you are using in the lab, usually your 96, 384, 1536 well plate, and we configure the Cellenion for you so that you can just adopt scale up your workflow using our device. You can use any sample type, any cell type, any sample volume. And because of our proprietary dispensing technology, which is very gentle to the cell, we have the best clonal outgrowth solution on the market. Here on the left, you see a platform that we've developed together with Biosero and which is powered by Green Button Go, GBG. It contains, so this Cellenion here, the Cellenion HT, inside which the single cell isolation occurs. The Cellenion is equipped with a shuttle here that extends to the right, from which sample and target plate can be inserted automatically using this PreciseLex 400 arm. Of course, this is only one configuration. More configuration can be developed. There's an incubator there that contains the target plates typically. The robotic arm will take target plates from this incubator and brings them to the Cellenion, and then once the isolation is finished, this will go back automatically there. Again, as I said, it's all controlled using GBG, and you see the control station here. What is unique about the Cellenion for automation? Well, first, sample loading. The technology that I will show you after is inherently suited for exchanging sample. So you don't need an operator to do that. The sample can be placed anywhere. It can be even brought here on the shuttle plate and loaded automatically by the Cellenion. We have a lot of automation method developed there. The dispenser is cleaned automatically. The dispensing QC is done automatically. We have error recovery methods so that it keeps on running or it recovers by itself. basically what you can do with the Cellenion 1 is to go from any number of samples to any number of plates. And finally, we have developed a very comprehensive API, and Dustin will show you more about that later also, that allows you to have full control of the Cellenion 1 over a third-party system like GBG. So what is it good for? You can simply scale up any existing single-cell workflow with it. It's what we believe is the only true walk-away operation for high throughput single-cell isolation, and it has a very low or no setup time because of the automation method that can even be scheduled. So you can come in the morning and the platform is ready for you to use. Now let's zoom in our technology. So if I stop a moment here you have the glass capillary with which we will produce drops containing cells. It's placed in front of a microscope here at the back. You see the microscope objective here. And what will happen basically is that we take a microscopic image of the tip of the nozzle. We will process this image to determine if there is one and only one single cell of interest inside the tip, inside the next drop. And if this is the case, the robot will move and dispense very gently this drop in the well of a microtiter plate. The second main great benefit of this technology, this glass capillary, is that it can aspirate the sample. And here, at the start of the video, we see the nozzle plunging inside the sample, the cell suspension being aspirated and placed back in front of the microscope. Here it's aspirated from an Eppendorf tube, but you can aspirate from any sample container. Once we're in front of there, we will take an image. The green box here corresponds to the volume of the next drop to be dispensed. When a single cell will be found in this green box, the robot will move and dispense the cell in a well. When no cell is being detected, the drop is recovered in a recovery tube placed underneath the microscope. So first cell is found, robot is moving, cell is dispensed in the well. And we come back and we repeat this process again and again until the full plate is being plated, basically, with single cell. When a drop is empty, or when more than one cell is detected, the sample is recovered in the recovery tube. There is no wastage, and we know that it's important for some B-cell cloning application where sample is really scarce. All images are recorded, which gives a full traceability of the workflow. So every time the microscope is taking an image, this image is recorded. Then you can have an image of your clones after the workflow is finished. You can sort out doublets or debris. It's not so important for B-cell cloning, but in some application we have debris using shape parameters. And basically you can go on and on like that at about 40 cells per minute. So that makes a 96 well plate in under 3 minutes and a 384 well plate in around 12 minutes. If you want to see a video of what's happening, so here is inside the Cellenion 1. You can see the glass capillary here placed in front of the microscope and the target plate here. And at the bottom here, you have a video of the capillary. The capillary is turned at 90 degrees compared to what you see here in real life. And on the left of the green bar here, you have the next drop to be dispensed. You'll see that when a single cell is detected in this volume, the robot is moving and dispensing. This is a real-time video, so it gives you a good impression of the throughput of the device. We will see an example where a doublet is coming. In that case, here is detected because the morphology is not the same as a single cell. So this is recovered underneath the microscope here in the sample reservoir. Until the plate is finished. Now, when we work in very high throughput, when we work with Biosero, we work with workset that contains more than just the Cellenion 1. It contains, for example, an incubator here from which a robot will come, the robotic arm will come and grab the plate. In this particular configuration, the plate is brought to a barcode reader, barcode is read, and then it is fed in the Cellenion 1 for single cell isolation. The lid is removed. The plate moves in. It is prepared by distatic. This is to ensure best precision in small well. and then single cells are isolated. Once the isolation is finished, the plate is given back to the robotic arm, it's releaded. In this particular workflow, it is brought to a centrifuge for a centrifugation step, and back in the incubator. Next plate is taken out of the incubator, and this process is repeated again. In terms of B-cell cloning workflow, what typically happens in the lab is that a number of samples are received in the morning, and they need to be prepared. PBMC separation and B-cell panning needs to occur before single-cell isolation. And what happens typically is that if in this case here, I've taken a scenario where there are 5 samples from 5 different animals that arrive in the morning. For each sample, we have between 5 and 50 microliter, around 200 cells per microliter, and a dead volume of around 2 microliter. In the work cell here, we have an incubator with 4 384 plates per sample being prepared. So we have a total of 20 target plates, but this is just one configuration for the sake of this talk today. And what will happen is that when the first sample will be ready, it will be brought by the operator or by the scientist inside the Cellenion HT, and the automated method will be started. The sample will be resuspended, loaded in the Cellenion HT, and the first target plate will be brought to the Cellenion HT. For each sample, we have 4 plates. The first plate, the Cellenion HT will start to plate on the first plate, while the scientist is preparing sample number 2, 3, 4, 5. When sample number 2 is ready, probably we won't get through the 4 plates here before sample 2 is ready. The operator simply pause the run, pause this method, insert sample number 2 and say to the machine, or say to GBG, sample 2 is in the instrument now, and resumes isolation. Isolation will resume with sample 1 until we've gone through these 4 plates, and once sample 1 will be finished plating, the Cellenion HT will wash itself, resuspend sample 2, and start taking the plates corresponding to batch number 2. And so on and so forth through these 5 samples here. We have a temperature-controlled sample holder here for best results. In terms of timeline of the workflow of Cascade, it looks like that. So in the morning, you can schedule the startup. So the Cellenion is ready when you come in. When the first sample is ready, you put it in and you start the workflow. Sample is being loaded by the Cellenion, so resuspended, aspirated. First plate comes in, single cell isolation occurs. The plate is sent back to the incubator, second plate, third plate, fourth plate. And then at the end of the fourth plate, we have an automated Cellenion wash, which cleans the nozzle, and the next sample can be processed. So basically, the Cellenion HT will process through samples autonomously. There's no need of operator to say, okay, now you do second sample, now third sample, or so on and so forth. It all happens automatically. Of course, this it did not only happen like that. We had to develop a lot of method for that, a lot of automation method to ensure and optimize uptime of the device. And I want to focus here on 3 of them, but there are many more that can be developed thanks to our very comprehensive API. First one is, we heard from the field, we heard from people using the Cellenion HT or people doing, scientists doing B-cell cloning, that they never know how many cell or how much cell sample volume they will have. Sometimes they have 10, sometimes 12 microliters, sometimes 20, sometimes there are only 8, and they can't really measure it neither. So when they bring it to the Cellenion HT here, they want to recover as much as possible. So if they have 10 microliters, they want to take 9 microliters. If they have 9, they still want to try to take 9 and see if it works or not. And this will not work, of course, if you try to aspirate more than there is in your sample, and you will have like a gap of air at the end of the capillary. This is picked up by the Cellenion. And what this recovery method does then, that it goes back to the sample reservoir, flushes back the 10 microliters that the scientist wanted to aspirate, and tries to aspirate 9 instead. And it will do that again and again until successful aspiration. And like that can enable the single cell isolation. So you won't need an operator to correct for this volume error. A second method, maybe, is this one here in the middle. So same story. Sometimes there are 200 cells per microliter, sometimes 50. Sometimes there will be enough to do 10 plates. Sometimes there will be only enough to do 2.5, but we don't quite know when we start the workflow. What the Cellenion does is that when it doesn't find cells for a while, it sends a flag to GBG, to Green Button Go, to our sister company Biosero, and says, it's a while, I've not been seeing any isolation, there is probably no more sample in the capillary. And then GBG can take the decision to stop this run and watch the Cellenion while it takes this plate away, skip the last plate or the last plates in the batch, and move on to the next sample instead of getting stuck somewhere. Last method here that I want to focus on is in case there are cells left in the capillary. So say you wanted to do 4 plates, you do your 4 plates, fine, and then you realize that or you don't realize, but Cellenion realizes that there is 6 microliter left of your sample in the nozzle. And this sample is very precious for you. What it will do, it will go to the sample reservoir and flush its 6 microliter back so that when you come in the morning, you still have this sample for other analysis if necessary. Here I presented 3 methods. There are much more that can be developed. And I think this is a good point where I can give the presentation over to Dustin, who will introduce you to the magic of GBG.
Very good, thank you, Matthew, and perfect. So all this automation that we've been discussing so far is of course powered by Biosero's Green Button Go software. We're seeing a bit of an example of that here, what the look and feel, the user experience is like here. So we're seeing an example of the layout tab here on the screen now, and this is where you would go in and replicate your work cell in the software. Once that's been built out, you go here to the process tab now, and this is where you actually build out your workflow, the work steps you want the automation to execute. And so we'd like to say that if you can talk through your workflow, you can actually build it in the software here with this low code, no code, drag and drop interface. So this workflow being built here now takes a plate from the hotel, goes to a de-sealing device, to a dispenser, back to a sealer, and then back to the hotel. And it's really that simple to build out a workflow within the software. Here we see the schedule tab, and this is where you can actually go in and calendar runs to execute at a given time and date in the future. So that, I mean, you can imagine it's an automation platform, of course. You might want it to execute overnight when no one's in the lab or over the weekend to really get that lights out automation and maximize the utility of your work cell. You also get some real-time monitoring capabilities. So what we're seeing here is an example of the run tab. This is typically what the user would see while a run is executing. So on the left-hand side of the screen, you see various instruments turning green as they're in use. If there was any sort of error or other condition, you might see a red color pop up, for example. On the right-hand side of that window there, you see a live Gantt chart being built out on the fly so that the user can easily walk up and see exactly what step we're on currently and what steps each plate has undergone throughout the course of this run. Next slide, please. And yes, here's an example that I built out of what the workflow that Matthew's mentioned so far might look like within the software. So we have the Cellenion feature here. This is an example of maybe something that the Cellenion HT platform could execute. So in the middle section of this window here, we see a plate coming from an incubator, going onto the Cellenion device, undergoing that centrifugation step, an optional barcoding scan step maybe, and then going back into the incubator. So this is kind of what a typical workflow could look like utilizing the Cellenion HT. On the right-hand side here, we see an example of the driver commands that are baked into our GBG driver for the Cellenion. So a lot of functionality is baked in and ready to go out of the box to get that fine control of the device that we want to see. Next slide, please. So working with our colleagues at Cellenion, we've developed an example method here that I'd like to show you. So what we're going to see here is a method that's designed to parse out a run definition file and then grab some parameters from that for a batch of plates or multiple batches of plates. And so we're seeing an example of that here. So you see 2 different batches of plates, each with their own associated set of parameters. For example, labwares, volumes, nests, being assigned on the fly from that run definition file, specifically for that batch. If we go to the next slide. So Matthew also alluded to some automated error recovery and error handling. And so the Cellenion driver utilizes Biosero's sequence driver. And so that's where we really get the fine control, where we can list out a series of conditions and evaluate them on the fly in real time and decide exactly what we want to do to achieve that high walk-away feature, walk-away time that we want to see with these automation platforms. So that's what we're seeing on the left-hand side there. A very powerful tool for achieving walk-away time. On the right-hand side here, we also have a live status monitoring window that comes up during this method to show exactly what's happening on the Cellenion, its status, what step it is within that sequence, and then what the plate barcode is in real time as a plate is undergoing its dispensing on the device. And so if we go to the next slide, we'll see this method in action and see some of these features. And here's the video playing now. So this is what the user would be seeing once this run starts up. They're prompted to select that definition file that I mentioned. We see those parameters being parsed in real time. So here's batch 1 and its associated set of parameters. Same thing for batch 2 here. We see those values, those variables being assigned. We see a look here of the storage plate contents within the incubator. And now the run has started with those parameters defined by the definition file. Of course, it's very fast in simulation here, but we see that live status monitoring window pop up as the plate is undergoing its dispensing on the Cellenion device. And then also, of course, this live Gantt chart being built out for each plate that's being run during this method right here. And so this will finish out very shortly, but just to highlight again, the real-time monitoring features of the Green Button Go software. So let's go to the next slide. And stepping back, just Biosero in general, I would say that we have a solution for the full spectrum of laboratory automation needs, starting from maybe a very small platform, maybe with one device, a robotic arm, and maybe some plate storage, just feeding plates to that device to get more walk-away time, more value-added time for the scientist in the lab, all the way up to maybe a much larger work cell, like in the bottom right here, where we have many devices, maybe a liquid handler, maybe even more than one robotic arm. These work cells are all powered by our massive device driver library of over 400 drivers at this point. This is all accomplished within Green Button Go Scheduler. However, if we go to the next slide, I'd like to also show you what can be capable within the Biosero family of software. So this is an example of a biofactory level automation, a full automated laboratory, if you will, going beyond the automated work cell. This is powered by Biosero's orchestration suite of products, the orchestrator level, if you will. And so this is comprised of many different work cells, each powered by their own instance of Green Button Go Scheduler, but all automated and all controlled, orchestrated, if you will, by Biosero's orchestrator family of products. So not only are plates coordinated, the plate movements coordinated between work cells, but also the data associated with all those samples and all those plates. Transportations are coordinated and seamless handoff of any plates or samples, labware, for example, from work cell to work cell to really automate the entire laboratory beyond the individual work cell. And so with that, I will hand it back to Matthew to wrap up.
Thank you, Dustin. And before wrapping up, I want just to take a time to emphasize the flexibility of the Cellenion and its main unique selling point compared to other instruments on the market. We have a sample management strategy that allows any throughput to be reached for any type of workflow. I gave you the example before of this B-cell cloning strategy where the operators bring one sample at a time inside the Cellenion HT. And before closing, I want to show you two more sample management strategies. Here in this slide is one of them where the sample is contained in a microtiter plate, which is in the incubator. So in that case here in the incubator, in this particular example, we have one sample plate and 20 target plates. And what will happen is that the robotic arm will take the sample plate, bring it to the Cellenion will resuspend the sample, load the sample, the plate will be brought back to the incubator, and the first sample will be dispensed, in that case, in the 4 next microtiter plate. When these 4 plates have been printed, the sample is washed, the same one is washed, the sample plate is being brought back, second sample is resuspended, aspirated, and the second batch is being processed, so on and so forth, until there is no more target plates or no more sample to be processed. Another strategy, another very clever strategy, which is used by some of our customers in monoclonal cell line development, is to place the sample in the well A1 of the first plate of a batch. So here you have the blue batch here of 4 target plates. The first plate of this batch will contain sample 1. When this plate will be fed in the Cellenion 1, sample will be resuspended, and single cells will be isolated from well 2 to well 384 here. The plate will be taken out, next plate will be taken in, and the 384 wells will be filled with a single cell. So on and so forth until the 4th plate. At the 4th plate, Cellenion 1 will wash itself. The first plate of the second batch will be brought in, and again, this contains the sample number 2 in A1, will be resuspended, loaded, and so on and so forth, again, until there is no more sample or until there are no more targets. We've had a lot of successful stories to tell with Biosero. One of them with a big pharma customer, where the Cellenion 1 came to replace an existing equipment, FACS, and where we had seamless integration. So this work cell had incubators, liquid dispenser, imager, and Biosero just took our Cellenion 1 HT and integrated it there in an existing work cell. And this was fantastic because the customer reported an increase in the throughput from 10 to 100-fold compared to what it was used before. Finally, today we talked a lot about B cells, but of course you can use the Cellenion 1 HT and Biosero for any type of integration, and Cellenion 1 HT for IPS or CRISPR cloning, for example, or for bacteria or yeast. We are very good at detecting single bacteria at the tip of the nozzle there. You can also use it for single-cell omics, single-cell whole genome sequencing, RNA-seq, single-cell proteomics. We can do everything. If you're interested, you can contact us for a demo and visit Lyon. We have this system here equipped with the Cellenion 1 HT and Green Button Go on site. The key takeaway message here is that if you take the Cellenion 1 HT compared to what you are using now, you will isolate more cells with less effort. What we see from the workflow of our current customer is that they reduce their FTEs for the sorting of cells by about conservatively 5 times, while increasing the throughput over 2 times. So they get 2 times more cells with 5 times less effort, and these are conservative numbers. With that, I would like to close the talk, and we are welcoming questions.
Hey everybody, I'm just going to hop back in and share some of these questions. So I see there's 2 questions in the chat. If anybody has any other questions to add, though, please feel free to drop them in now and we will make sure to discuss them. So Matthew, I think this first one is for you. Can the system tell the difference between live and dead cells?
Absolutely. So the system is equipped with fluorescence. So you can have like this usual green-red marker for live dead cells, and you can therefore sort only live cells if you want. What we've also seen is that some researchers, more than pharma companies, have looked at the morphology of cells and tried to correlate the morphology of the cells with whether they will give out clonal outgrowth or not. But it can be done also the conservative way using a green dye.
Okay, next question. Does the system work with samples in 96 well plates?
Absolutely. It can work in any, the sample can be placed anywhere. In the 96 well plates also, although it will be a large volume for what is really necessary, but it can also be done in 96 well plate.
Awesome. Okay, so I haven't seen any other questions come in. I'll give it just one more second to see if anybody has anything to add. No, I think that's bringing us to the end of our time today. So with that, I'd like to thank Matthew and Dustin for presenting today, and for everyone who joined us online. The webinar recording is going to be available immediately after we wrap up. So if any of us can help with your automation needs, you can find us online at cellenion.com and biosero.com. Thanks for joining us today.
Thank you very much.
Thanks, everyone.